universe. Anything you see is made up of something else, which is smaller than the original thing. Basically, the universe is divisible. Even you are divisible. You're made up of organs, which are made up of body tissues, which are made up of cells, which are made up of organelles, which are made up of molecules, which are made up of atoms, which are made up of protons, neutrons, and electrons, which are made up of quarks. However, in this documentary, we're not going that far. Let's look into cells, as it's easier to explain biology than it is to explain quantum mechanics. Or at least so I hope. So, let's run a refresher in history. In 1590s, a person called Hans Jansen and his son Zacharias built the first microscope ever in Cork. Some years later, in the year of 1665, some other dude called Robin Hood discovered the first cells, leading us to having to study about them. Don't you just love it when some random freaking dude? However, the first theory is credited to Theodor Schwann and Matthias Jacob Schleien around the 1830s. The different cells organelles that compose them were discovered by a lot of different people throughout the years. The nucleus, for example, was discovered in 1833 by Robert Brown, and the ribosomes were only discovered in 1955 by Gerard Claude. And Maurice Wilkins, Francis Crick, and James Watson discovered the structure of DNA in 1953 by taking X-ray photographs of molecules. Okay, so now that you know about the culprits for having us to make this video, let's start by asking, what are cells? If you don't know that, either you really haven't been paying attention to your biology lessons, or you actually need medical attention for undiagnosed Alzheimer's. But alas, we're gonna say it either way. Cells are the basic building blocks of all living things. They're these really tiny blobs that work like Legos to build bigger things. What about the cell theory that the teacher asked us to cover here? Well, cell theory is a concept that has been universally accepted that mentions that living things are all made of cells. Now, that was very simple. Let's complicate it. Cells differ accordingly to different living things, but sometimes they have the same components. For example, vacuoles. They can be found in any animal and in plant cells. Let's start with the organelle that compose animal cells. Starting off with the basics, the nucleus. The nucleus is a part of the cell that contains all of the genetic material needed, expressed in DNA. DNA is short for deoxyribonucleic acid and contains the genetic code of organisms. DNA can tell you what proteins to make, and in case you didn't know, protein basically do anything in your body. It's known to be a double helix of nucleic acids, a type of macromolecule. These nucleic acids are made of phosphate group, a sugar group with 5 carbons, and a nitrogenous base. When replicating, DNA is semi-conservative because it replicates by creating 2 new strands of DNA and combining each of them with one of the old ones. Helicase separates from the DNA strands. DNA polymerates create 1 new strand for each old strand. However, it only reads DNA from the 5th prime to the 3rd prime. In case you didn't know, the number of carbons in the sugar base is called a prime. So when it encounters DNA that goes from the 3rd prime to the 5th prime, it has to take breaks instead of reading continuously. So, with DNA explained, let's go back to the nucleus. The nucleus also controls how genes are activated, basically controlling the protein production and therefore its activity. It is surrounded by the nuclear envelope, separating it from the cytoplasm. In addition to that, the nucleus also has a nucleus, which is where ribosomes are made. But let's focus on that protein production aspect. In order for DNA to work, it needs to make the protein coded into it. For that, the process of transcription and translation need to occur. During transcription, an enzyme called RNA polymerase breaks the DNA nucleotides and makes mRNA, or messenger RNA. This mRNA, unlike the DNA, can leave the nucleus of the cell, so after being made, the mRNA leaves and goes into the cytoplasm where it finds a ribosome. But what's a ribosome? Ribosomes are another type of organelle. They're the ones that synthesize the protein. Okay, now that it has received the mRNA, translation starts. Ribosomes are made of rRNA or ribosomal RNA. They will read the mRNA to make proteins. In order to do that, they use the tRNA or trans-RNA. tRNA is the type of RNA that carries an amino acid with it, so the ribosomes will connect the tRNA antibodies with the mRNA codons. Codons are 3 nucleic base pairs in the mRNA, and anticodons are 3 nucleotide base pairs in the tRNA. When the codons connect, the tRNA will leave behind its amino acid with the ribosome, which connect to the other amino acids to make an amino acid chain. This chain then goes to the ER, or the endoplasmic reticulum. It produces, processes, and transports protein that were synthesized by ribosomes. Here, they take the chain and turn them into full-fledged proteins. There are two kinds of ER, smooth and rough. The rough one has ribosomes attached to its surface for easy protein access, and the smooth one does not, and instead makes lipids and hormones and removes toxins. Okay, so let's talk about the Golgi apparatus. It consists of membrane brown sacs that originate from the vesicles that came from the ER. The Golgi apparatus receives the proteins processed by the ER and puts them into vesicles.
Like little packages. Also, the Golgi apparatus always appears to chain the endoplasmic reticulum.
The mitochondria has as its function the energy production of the cell, and that is why it's very known to be the powerhouse of the cell. And that is where the process of cellular respiration takes place. Cellular respiration is when sugar and fats, two very important macromolecules, are broken down, releasing energy in the form of ATP or endosome triphosphorate, which is basically a very strong battery. So, we've been talking about macromolecules a lot, but what are they? They're important cellular components and perform a wide array of functions necessary for the survival and growth of living organisms. The four major classes of biological macromolecules are carbohydrates, like the sugar the DNA and the one used for mitochondria, lipids, like the one made in the smooth ER, proteins, like the ones made by the ribosomes/ER, and nucleic acids, like the one in the DNA. Macromolecules, of which we have studied, have been one of the components that were essential for the functioning and creating of life way back in time, and they are still super important in maintaining what scientists believe they created: life. If you don't want to know more about the production and about the roles, you can skip to this part. So, proteins, they work in transport, storage, or membranes. Each cell in the living system contains thousands of proteins. They're made by the ribosomes and by the ER. Nucleic acids, like the DNA and the RNA, are used as information. They are generally made by using amino acids, which we ingest when we eat proteins. Lipids are compounds that are largely non-polar in nature. Cells store energy for long-term use in form of fats, and they also provide insulation for the environment for animals and plants, usually made in the smooth ER. Carbohydrates, they provide energy to the body. They also contain fibers, which promote regular bowel movements by adding bulk, generally eaten by most animals, although some living beings make them, such as plants. Now that we know about each of them, let's learn more about them generally. They are formed by smaller molecules called monomers when organized in a specific sequence. After being made, the Golgi complex is responsible for its secretion. So, now that you know about macromolecules more, you can learn about what recycles it. Lysosomes are a type of vesicle that contains digestive enzymes that break down large molecules such as organelles, but they're also able to break down macromolecules usually used to repair cell membranes. Typically, lysosomes are the recycling system of the cells. Okay, so I should probably talk about the house of the organelles. The cytoplasm is under the cytosol in the cells, plus the organelles in it. Except for the nucleus. The cytosol is a gel-like liquid that consists mostly of water, but also has ions, proteins, and other small molecules. It has a pH of around 7, which, it's expected because it's made of water. I have a question for you guys. How do you think the organelles stay in place inside of the cell if it's floating around in some kind of hair gel? Leave the answer down below in the comments! Nah, I'm just kidding, it was a rhetorical question. The answer is the cytoskeleton. There are filaments and tubules within the cytoplasm that have different functions. There are 3 cytoskeletal filaments types: microfilaments, which help with the movement of the cell from the outside, microtubules, which help with transport and movement of the cell from the outside, and the intermediate filaments, which give structural support to the cell and keep it in place.
Hey guys, editor here, and on this scene, the narrator had a stroke and wasn't able to pronounce phospholipids. So yeah, keep in mind that if you hear a word that makes no sense and that doesn't exist, it's probably phospholipids.
Finally, for the animal cells, we have the cell membrane. It's made of phosphophylase, molecules with polar and apolar sides. When together, phosphophylase form a phosphofolio bilateral because the hydrophilic and hydrophilic properties of their heads and tails, phospholipase and lipids. This is basically what makes it most impermeable and separates from the outer environment. However, certain things can pass through it. Oxygen and carbon dioxide pass through it easily in a process called diffusion, where they literally just phase through it, while larger or charged molecules have to do either facilitated diffusion, where hydrophilic molecules pass through a hydrophilic channel, or active transport, where ATP is used to make proteins squeeze through something. There's also osmosis, where water goes in and out of the cell to balance out the amount of diffused things internally and externally. Last but not least, there's also the exo or endocytosis. Exocytosis is when a vesicle fuses with the membrane and lets its contents out. It can be used for cleaning. Endocytosis is when a vacuole is formed to eat something and take it inside of the cell. It can be used for eating, but it's also used by the phagocytes to do phagocytosis and kill things like bacteria. Talk about similarity and difference between plant cells and animal cells. They share the common components such as the nucleus, cytoplasm, mitochondria, and the cell membrane. However, plant cells have 3 extra components: a vacuole, chloroplast, and a cell wall. The cell wall is the part that surrounds the plasma membrane of plant cells and provides protection to the other organelles and provides structural support. It also allows cells to develop Tugger pressure, which is the pressure of the cell contents against the cell wall. Vacuoles are really important to maintain the homeostasis of the plant. It is involved in the control of cell volume by filling up and emptying itself, the regulation of cytoplasmic ions and pH, storage of amino acids, sugar, and CO2, and the segregation of toxic ions and xenophobics. Basically, it cleans stuff up and stores stuff. Chloroplasts are organelles that convert light energy from the sun into chemical energy with this photosynthesis process. They sustain life on Earth, being the starting point for the energy all living things use. Chloroplasts also provide diverse metabolic activities for plant cells, including the synthesis of fatty acids, membrane lipids, and others. In conclusion, life is something so complicated that we spend hundreds of years and billions of dollars studying only their tiny bits. Regardless, every microscopic living block that makes up this giant ecosystem is essential to the maintenance of life. So, next time someone's giving you a hard time, just remember they're made of really small and goes think of yourself as a giant Lego-man because, well, I don't know, it's hard to be inspirational. Just deal with this, it'll be funny. Anyways, thank you for watching.
Hi, my name is Ana Marina and I helped in the biology midterm in the part of information, more specifically at the part of cells history and macromolecules to make it easier for Diogo and Davi to do the script. Thanks for watching.
Hi everybody, Bruno here, and I really like to do the biology project because it's really like working with friends and working collaboratively, and I really liked, and I mostly helped in the part of getting information. Thank you, Ms. Marcia, and bye.
My name is Davi, and I helped with doing the editing and script for this project. Also helped a little bit with the research, talking about specifically what topic that I choose to help with the research, how do processes in the cell wall happen.
Hey guys, Diogo here, I'm the second editor, and I did half of the scenes together with Davi, and also am editing the final video, so I joined the scenes together. Also, I'm the supervisor for information, so I double-checked everything that we did, and edited information every now and then.
This is Karen, the person in charge of narration, who also helped out a bit with the script and research. Thank you so much for watching our video once again, and I hope you enjoyed it.